Hello, everyone. Welcome to today's webinar, Downstream Process Optimization with Synthace. My colleague Lucy and myself will be presenting today, and we'll be talking about how to make the most of scale-down, high-throughput process development tools in different stages of your downstream process optimization campaigns and how Synthace can help along the way. Now, if you've already attended a webinar of ours in the past, welcome back. If you're joining for the first time, it's great to have you. Can't wait to share the session with you today and hear your questions as well. Just a brief introduction. So myself, I am Victoria González. I'm a product manager here at Synthace. Prior to that, I was actually in the customer success team, setting up Synthace for purification, process development applications with our client base. And I have a background in cell biology and life sciences in general. Lucy, do you want to introduce yourself as well?
Yeah, sure. Hi, my name is Lucy. I'm a senior scientist here at Synthace, and I have been part of developing the products we are going to showcase to you today that will hopefully help you with your downstream optimizations.
Thanks, Lucy. So I will just also give you a little bit of an overview of what you're going to be hearing about from us today. So first, I will start by talking a bit more generally about downstream process development and the challenges that we encounter there and how Synthace can help address those, and also particularly focus on high throughput process development tooling. I will also give you a brief overview of the Synthace platform, and I'll hand over to my colleague Lucy, who will walk you through how to set up actual applications with Synthace. On to the first section of our webinar, so where I will talk to you a little bit about the challenges of downstream process development that I'm sure many of you must be aware of, which is probably why you're here with us today, and how to apply high-throughput process development tools. But before we get into the weeds, I was actually hoping to get a pulse check from you. And so you should be seeing a quick poll. If you could take a minute to answer about whether or not you've used high throughput process development tools in the past, and if yes, which ones? So I see this quite a spread. Some of you are new to the tools, which is exciting to see. Hopefully, we should be able to give you some tips, encourage you to try them out. And I can see that there's quite a split in the audience between the different tools because there's quite a range of them available. We're here today to talk about a downstream process, and of course, as illustrated on this example, that is about monoclonal antibody purification, but it can be extended to other types in the business of recovery of biosynthetic products, which is what we're concerned with in downstream process, right? When you will get harvest from the upstream process, from the bioreactors, they will typically go through several stages within the downstream process to try and obtain as much of the product of interest in as pure a form as possible, right? And typically this process will involve several chromatography steps, maybe of different types, such as protein A, ion exchange, hydrophobic filtration, which need to be adapted or even fine-tuned to the actual compound of interest that you are trying to obtain during downstream process development. And considering the increasing complexity, variability, high quantities of product generated in the bioprocessing upstream pipelines today, there certainly are challenges associated with this, as I'm sure you will be familiar with. So I've listed some of these challenges here. First of all, of course, there's no one-size-fits-all downstream purification approach. There may be general rules that you can follow, but in the end, there's nothing better than an experimental approach to actually test them out and see if you're obtaining the results that you expect. Downstream process development experiments are costly and they're pretty resource intensive, both in terms of samples and materials that you need to invest, but also time that you need to invest to actually set them up. So those are typically multi-hour runs requiring quite a lot of materials. In these experiments, there's also actually a really broad, multidimensional design space that you need to probe to identify these optimal conditions. There's so many variables, so many parameters that you can be tweaking to get to that optimal purification process. In those parameters, even minor inefficiencies in the process, once you move on from process validation and development through to scale up and actually usage in production manufacturing pipelines, even minor inefficiencies can result in big product loss, therefore big loss in terms of also income, right? And a risk to the production pipeline. That's why high throughput process development tools have really become instrumental in making process optimization more efficient. And that is because these typically scaled-out miniaturized tools allowed to test many conditions in reduced volumes. This means, of course, cost savings. They're also typically amenable to automation, so they will allow you high throughput experiments with gains in terms of walkaway time and reduced potential for introduction of errors and so on, particularly relevant when you're testing many conditions. They're also compatible with design of experiment approaches, which, as we'll explain later, you may wish to consider within your downstream process optimization campaign to probe more of the experimental space more efficiently. Even though these products don't typically have inline analytics that you can obtain directly, they will, however, output samples usually in a plate format that is easily amenable for high throughput analysis. And so you can obtain quite a lot of data pretty quickly. Of course, there may be considerations in utilizing these high throughput process development tools, which we will touch on when we discuss the different tools that we've listed here. But overall, they do show nice correlation with scale-up models and can be used to learn a lot about your system early on. I just wanted to list some of these tools that we have interacted with quite a lot at Synthace and with our customers. These tools are good for different sorts of applications. So there's really a different tool for each purpose. For example, filter plates, which are micro-target plates where each well contains a resin or filter matrix and typically also a filter membrane. These plates have an aperture at the bottom, right? So that the liquids can flow through while the resin remains. The liquids will typically flow through under force, such as positive pressure, negative pressure, or centrifugation. So this type of purification really is a batch format where each separation occurs only once, but interaction between protein and resin is pretty much the same or equivalent to column formats. There's many applications for which filter plates are really very well suited, such as, for example, screening of many different resins or different buffers, and also applications like measuring isotherm. Some of the benefits of filter plates really is that they're really cost effective. They come in this plate format that can be, for example, 96 wells, allowing you to assess 96 conditions in parallel. And within a day, within a single run, you can be dealing with several of these plates, and therefore it is really easily scalable to many conditions, many samples. And it is also compatible with a variety of both automation setup, but also even manual setup. Of course, they do have some considerations. So it is, as I was mentioning, batch mode purification. So it may be a little bit further from the scale-up model of column chromatography. But overall, it does allow you to assess quite a lot already, thanks to its plate format, really in a high throughput manner. So for early stage screening. Another type of tools are RoboColumns, which are small packed bed columns available in a range of bed volume, so from 50 microliters to 600 microliters. Unlike the plate format, so they do represent a scaled-down model of a chromatography column, and they're available either pre-packed or with use-defined resins. And so these can be used for applications like optimization of purification conditions of many types of conditions, right? In this case, for example, you can also start optimizing things like residence time, since it is in column format, but also buffer conditions, etc., and also dynamic binding capacity studies. And some of the strengths of this type of tool, as I was mentioning, so it does come in a column format, so it's closer to a scale-up model. In this case, you can be doing 8 conditions in parallel, and this is usually limited by the way that the steel tips that are required in combination with this RoboColumns are set up on a liquid handling arm. So you'll have 8 in parallel, but overall on one of these RoboColumn plates, you can fit up to 96 columns. So you can be doing up to 96 conditions in a run. Using RoboColumns, you can start mimicking a more complex elution, like gradient elution. So by generating like a step gradient and mimicking sort of a pseudo gradient elution. And so the RoboColumns are automatable. So they're automated on Tecan liquid handling platforms. And now some considerations to mention with RoboColumns is that there may be, of course, it is a small column, there are some limitations in terms of separation resolution. And even though it is mimicking flow quite nicely, it's still going to be interrupted flow depending on the size of syringes that your liquid handler is equipped with. So it may only be able to transfer 1 milliliter in one go or 5 milliliters in one go, there will be necessarily some interruptions in the flow. But still, it's a very good scale down model. There's also other tools that are certainly worth mentioning. So for instance, resin-filled tips, very popular, or also magnetic beads. And they're really amenable to a range of applications. If for high-throughput purification, particularly good for eluting in small volumes, so pretty concentrated. And of course, they're also easily scalable. And it is possible to go through hundreds of conditions of samples per run. And they are also compatible with a variety of automation manual setups. So we won't go too deep into these tools today, and just maybe worth mentioning some considerations. So they may be further, again, from the scale-up model of a column. With the filter tips, you do have bidirectional flow in the way that they operate. And with some of these, like with beads, you may be considering costs when you're thinking about scale of your experiments. Now, all of these tools can be used standalone in different stages of your process development campaign, but also they can be combined, right, for different levels of focus as you progress for your process development optimization study. So, for example, you may want to think early on to do a high throughput screening of many resin and buffer conditions and sample a very large space. So really test a lot of different combinations when you're not sure what to go with, and filter plates are an excellent tool for that. And then perhaps you may want to drill down in a part of that space that you've identified as potentially interesting or better suited to your product that you're trying to purify. And so you may want to use, for example, RoboColumns to explore more of the purification design space and more different conditions in a column format. And after this, of course, you may go into the next stage of process validation, for example, with an active system, so maybe with flow-based purification to really refine the method. And so like this, you can really make the most of high-throughput process development tools, conducting multiple runs to characterize your system, to fully understand your system, mirror the scale-up model as closely as possible, and really learn as much as you can about your system before scaling up. Now, setting up purification process development experiments with high-throughput process development tools does come with a few challenges today. So particularly considering more complex designs with multiple factors, multiple levels, it is complex enough to set it up. But additionally, scientists may be distracted by understanding whether this experiment is actually feasible and how to execute it, which may get in the way and limit what studies you are actually going to make reality in the lab. Using automation for such type of studies does require pretty complex automation programming. And once you've nailed one down and you've been able to describe one protocol, then to change it to the next study that comes your way is pretty difficult. So it can be pretty inflexible. And once you have been able to execute it, your experiment, you are then faced with a great task of actually wrangling all the data together and being able to then analyze it and extract insights. And so these are the type of challenges that we have looked to address with Synthace so that you can really make more powerful, more insightful experiments using these high throughput process development tools. And so that scientists can focus on the best possible design, utilize automation fully, make it universally accessible and flexible, and help also with rapid data structuring and visualization. And up until very recently, we were really focused on the RoboColumns model on Tecan platforms for this, and we continue to do so. But also now we're very excited to add the filter plates application to our portfolio, and so now we can help you with more steps of purification process development campaign, and that's what we would really like to talk to you about today. So now I would like to explain to you a bit how we will make it possible. I'll start by just giving a more general introduction to the Synthace digital experiment platform, and then hand over to my colleague Lucy, who will actually tell you about how would you go about setting up real experiments. The Synthace digital experiment platform covers the experimental journey of a scientist from the design and plan stage through to the execution in the lab with automation equipment or manually, and then actually wrangling and analyzing the data. The idea is that whereas in a typical experiment, the scientist needs to be concerned with preparing the experimental design, but also all the calculations and the low-level abstractions, keep track of samples, the experimental data, perform the analysis, and put all the record together. And it all lives in your head in a mental model of the experiment. With Synthace, we want to provide a digital experiment model that centralizes all of the parts of your experimental journey and makes it easily accessible, easily transferable, easily reproducible for scientists. So starting with the design step, here, scientists are able to define and plan their experiments in an intuitive interface, abstracted from device constraints and focused on actual protocol steps. If we look at it here, we have multiple blocks that designate different steps within an experiment, which is what we call elements. And all of them are easily configurable in kind of biology-friendly speak. So it's a bit like answering a questionnaire where you just fill out what it is what you would like of your experiment. I will tell you a bit more about it later. You can then actually simulate this experiment. So against your device, and the simulation will show you what the experiment will actually look like. So it checks the feasibility within the constraints of the setup, shows you how it's going to be executed step by step, as you see here, perform calculations of how much you need for each buffer, for example, and really takes care of a lot of that complexity for you. You can then use the instructions generated in the simulation, and they're transferred directly onto the device, where we also handle the execution. So as you see on this video, in this case, on a Fluent device, for instance, on a Tecan Fluent device, we take care of the actual execution and also collect the data off the back of that, for instance, plate reader data. And then brings me to my next point, which is analysis. So finally, we help scientists with data aggregation together with all that metadata collected from the design and the run. And for certain applications, we also actually perform a specialized data analysis, such as for a RoboColumn experiment, as I'm showing here. So when thinking about an automated experiment, scientists are typically tasked with translating their intention, their protocol that they're thinking about, into low-level instructions for an automated device. And in this case, as you might have faced before, we need to think about all the aspects of each liquid transfer from pre-mix through aspiration, dispense, post-mix on a well-by basis. And complex protocols may require hundreds of these non-intuitive instructions, and there's certainly tooling to make it better, but they do not contain the biological intent, typically, of what the scientists want to do. They do not reflect the design. And so instead, in Synthace, we've approached experiment design is with biology in focus. We have what we call elements, which are this kind of elemental building blocks, experimental building blocks that define simple actions where you can express what you would like to happen without having to worry about low-level extractions. Much the same like you wouldn't worry about it when you're thinking about a protocol that you would execute manually. You'd need to introduce, for instance, how much you would like to aliquot, how many replicates, and then the element would be translating it to automatable instructions under the hood. We have different types of building blocks, some pretty generic that allow you to do everyday actions in the laboratory, like aliquoting, making mixtures, diluting, etc. And also we have some that are specific to particular applications, so developed with, for instance, with purification in mind, as we'll see later. These elements then get put together, as of course every protocol, every method will have several of these steps to what we call a workflow, where it will typically have some inputs, like, for example, the type of liquids that we're dealing with, or the type of physical labware that we'll be operating with. And then we'll have several elements, several of these steps. And as an output, we'll generate the end plate, the end product, etc. For instance, here's a simple example of Bradford's assay to give you an idea of how it works. So here we've used elements to define our standards, our samples, and then other elements to actually perform dilutions, to replicate them, and to add a reagent on top of them at the end. But of course, hopefully this gives you a bit of an idea, but we're not here to talk about just simple experiments, definitely not the breadboards. We wanted to talk about experiments with the power and complexity required to truly understand, characterize the purification process. And that is what my colleague Lucy is going to talk about. So I am just going to hand over to her.
Thank you. So thank you so much, Victoria, for the great overview of the platform. I hope this gives you a better idea of how Synthace works. And now I will try to give you concrete examples how we leverage our platform for these specific types of applications where we try and employ these tools. As Victoria outlined, pretty much any downstream process optimization campaign would begin with filter plates, just because it's so flexible. It gives you so many runs in parallel. So you can go crazy. You can really understand your experimental space really well. And so pretty much as with any plate-based assays, you start thinking about what combinations of factors do I want to explore? How many wells am I going to need for that? And it's a lot of stuff you need to constantly edit yourself back on. You constantly need to iterate. How many wells can I cut here? How many conditions can I include here? And it's really taxing. And so what I want to really highlight here is the features that we bring as part of our platform. And so these first 2 points really speak to what I've just described, is the fact that you no longer have to think this way. Synthace takes care of that. All you have to worry about is really the variables you're trying to explore, and Synthace enables you to do this iteration much quicker. It helps you stop thinking about plate layouts all the time because it automates this for you. And also, in terms of programming these types of experiments on the robots, where you need to think about steps like loading liquids onto the plate, taking the plate to T-shake so that it mixes, all these things will need to be programmed manually, but Synthace really does this for you as it generates these low-level instructions. All you need to do is express it in a biological friendly way. I want this and this to happen. And finally, but last but not least, we also automatically collect all the data and help you with the structuring of it, which really speeds up the analysis. And I'm not sure if I highlighted it yet, but this feature is available on the Tecan Fluent instrument so far. And so going back to the elements that Victoria was speaking about, we have two specialized elements for this type of experiments. One is called the design filtration protocol. And that's really central to designing the entire filter plate experiment. It is here where you define what factors you want to explore. And this is what generates the overall design of the experiment. And then the second element, which is the run filtration stage element, this is where you define what liquids you want to load, how long you want to incubate for, how do you want to filter the liquid through the filter plate. And so going back to the example that Victoria alluded to, let's think of a standard monoclonal antibody purification buffer screens. So this is quite a typical diagram that I'm sure you've seen before. So let's imagine you have a protein A affinity resin in the filter plate, and you need to go through a series of stages. And these tend to be quite similar between the different modes of purification. So I'm sure you quite know what I'm talking about here. And so you start with washing the resin a few times. This all goes to waste. And next you add your sample, you incubate it for a set period of time of your choice. Then you filter that through the filter plate and collect it for later analysis. Then you want to wash the resin to get rid of any impurities. And finally, you want to elute your protein. Again, you apply the elution buffer 1 to 3 times and collect the eluate each time. And so imagine that the goal here is to, again, maximize yield and minimize, for example, host cell protein impurities. Like this is quite a standard process. This is very often the target with maybe some additional protein quality characteristics that you need to look out for as well. And so let's imagine this is going to be a pretty classical wash and elution study where we, for example, explore the wash buffer type. So what buffer we will be using for this wash step, and maybe explore an additional additive. whether it can help with decreasing this wholesale protein impurity load. And next, maybe looking at also elution buffer pH, because for protein A purification, we typically elute with a low pH. That tends to be quite detrimental to the protein. So maybe we can strive to increase it a bit. But as you can see here, we are looking at 3 different factors, but that can already, depending on the factor ranges you set, it can create quite a substantial number of runs. And so this is how it would look like in Synthace. So I know at first it can be quite a face palm a moment, like this looks complicated. But if you really focus in and drill down on what it does, it's really just the same element repeated multiple times, really speaking to the modularity of this platform. So in this first stage, it's this design filtration protocol element that I talked about. So here you're simply expressing what the actual filter plate experiment is going to be. In these repeated blocks, all you're doing is, again, separating out the protocol into the separate purification stages. So for example, here you will be loading an equilibration buffer, whereas here you will be loading your sample. So the liquids might change, the incubation times might change, but you can flexibly set this up. And then over here, you might be cleaning and storing your resin, but this is typically not done as filter plates tend to be discarded after first use. And finally, you want to plate weight all the collected fractions. And so the thing I want to highlight here is that you express what you want to happen, and then Synthace will take care of all the low-level instructions. So you don't need to worry about those. And I will showcase that now. So let's take a look at a live demo. Those never go wrong, right? So let's see how it looks like. So here I'm showing you a design filtration protocol element. And so you would start here with your design, where you express the factors that you want to look at. As I said, we will be looking at pH. We are also looking at wash buffer type, and we're looking at an additive concentration, which in this case is sodium chloride. And you simply have to express the levels you're looking at. So as you can see here, I've chosen pH and I want to look at these levels. And I've set up something similar for the others. Then in the liquids tab, we have to describe what liquids we will be using. And so you can see here that some liquids, we're not really interested in exploring them right now. So they're simply there to be used. So the equilibration buffer doesn't have any of these factors of light. We simply specified the metadata for this component, and it's equal to 1 liquid. In contrast, if we look at, for example, the wash buffers, here we are applying 2 of the factors. And as you can see, Synthace has automatically created combinatorially 12 liquids that will be needed for this step. And as you begin to appreciate, since here Synthace created 4 liquids and here 12, we will be requiring 48 wells. And so it's quite fast to see what will be the requirement, but I will show you shortly how it's even more visible. As here we've defined, we're going to repeat all those conditions twice. And then here in the filtration plate layout is where we describe what resins we will be using. You can see this entire plate is highlighted as protein A resin, but equally you could add different resin types with different volumes, and all that information is captured there. Whereas if you were to execute this manually using a script, all this information would be missing. And as you can see here, this is what I was alluding to. We have 48 factorial combinations and 2 replicates, which means you will be requiring 96 wells. And so I hope you can appreciate literally just staying in this element and switching between these 2 fields. You can quickly iterate and quickly understand what is feasible, what isn't, and quickly design your experiment. And next, I'll just quickly show you the run filtration stage element, which in this case I'm using for the load stage. You can see this will be repeated just once. And as I've alluded to, we have fields to set up what will be loaded, how much, then incubation conditions, how long do you want to incubate for, what temperature, what's the speed you want to be shaking the plate at, and finally also the filtration mode, what type of filtration mode you want to be selected. And last but not least, more options, that's right. You can choose whether you want to collect the filtrate, what type of plate it will go into, and whether you want a blank plate. So I hope you can appreciate that in this one small element, there's basically this will correspond to hundreds of low-level instructions for the device that will be automatically generated. And so once you're happy with your setup, all you need to do is click Simulate, and this will automatically compute the instructions, but I'm not going to wait just here. So I'll quickly show you how it looks like. And all you need to do is just literally step by step, have a look what will physically happen on the device. And I hope you can see that clearly here. There's really low level detail of what will be expected where and from where. And importantly, you get an overview of how much buffer you will actually need to set up this kind of experiment. So when you show up in the lab, all you need to do is set up the specific liquids in the specific positions, put empty plates where they are asked for. And when you're ready to run, all you need to do is hit schedule. And what this will do is send this to the computer in the lab where you just need to press play when you're ready. So that's clearly quite a lot of power from just being able to set this up at your desk. And moving on, I hope you could see that how much help Synthace can be in designing your experiment. But imagine you've executed it and it's a full 96 well plate and you've been collecting fractions. And so this is a screenshot from the run that we executed. And so you can see we've collected about, I think, 13 plates worth of material, which were all automatically read on the plate reader. And this actually translates to a lot of raw plate reader files. And so you can see here, and I hope many of you have seen these before, they don't really mean much until context is added to them. So you see you have an absorbance measurement, some well location, and some measurement here, but it's really meaningless until you provide that data context. And so you could almost argue that data structuring is really half the battle. And for this, we provide this functionality, which we call the experiment map. And so here we basically try to track this entire design, run, analyze cycle, although it looks a bit more linear here. This probably doesn't look like much to you, but what this really translates to is these parts of the experimental cycle. So here you have the design and plan parts, which are the workflow and the simulation parts, which I have shown you. This is the execution captured. So this will have all the data associated with the execution. And finally, you can add this additional app. So it's a functionality that Synthace provides, which will structure the data for you coming from this execution. So you will have the data, the raw data structured with the context from the experiment. And so you can see that here within this app, you have the raw measurements from the plate reader, but also important context from the experiment that will help you analyze your data later on in other software. So you see here, you have specific plates that these measurements came from, and also some experimental context that will definitely help. But what if you want to also just quickly see how the experiment worked? Do not be scared. We also provide some help here. So for this, we have the View Data app, which will give you precisely this functionality. And so even though it doesn't really look like much, this is very powerful as it gives you very quick overview. Has anything gone really wrong? Is there signal? Is there an absorbance in the places where I expect it to be? So for example, if you look at the elution plate, you can see here clearly we are getting a lot of signals. So we are successfully eluting protein, which gives you a quick understanding, right, things have gone okay. But going back to our story of going through a process development campaign, what we're aiming to do here, we are hoping to narrow down that experimental space. So I hope you can appreciate that we screened 48 conditions in duplicate. We were looking at these two factors, really. And running this experiment, which took maybe 3 hours, helped us understand that, okay, elution pH can probably be fixed to the lowest value. Unfortunately, there wasn't much help there. And the sodium chloride addition past a certain point wasn't really contributing much to the result. And so you can imagine running this kind of experiment and coming to these sorts of conclusions where maybe you don't have your amazing process already fixed, but it helps narrow down those factor ranges that you can explore further using RoboColumns. So this is our older product offering. And as I hope you can appreciate that even though in the picture 96 columns, it is actually only 8 columns that you can run in parallel, merely because of the fact that the liquid handler will only have 8 tips that it can insert in at any one time. So that basically, what that means in practice is you need to run 8 columns purification first until you can move on to the next one. And so once again, the features of RoboColumns is that this time you can also use it on the Tecan Evo instruments. But likewise, we still provide the same intuitive and flexible setup for every chromatography stage. We also support linear as well as pseudo-gradient chromatography, where all those gradient buffer mixtures that you may have to do manually otherwise are prepared automatically. The same, very similarly to the way it would work on an ACTA, where you simply define start percentage of buffer B and end percentage of buffer B. And finally, we also provide automated data collection, structuring, and application-specific analysis. So what this means is this analysis is very specific to RoboColumns, and it's specifically meaningful for that application. And finally, we also enable really fast cherry picking of fraction of interest based on this analysis, which I will show you briefly. And again, going back to the elements, we have some specific elements for this application also, where first up, you define what are the resins you're going to be dealing with. And so this is really important because you capture all that information related to the resin you are using for that experiment within the context of all the liquid handling instructions. Next, we have the define RoboColumn plate element. This is where you physically specify what is the column size, what is their location on the actual plate, and you can fill in what the resin is going to be that is being fed in from this element. And then our big stars of the show are the run chromatography stage and run gradient chromatography stage. They're quite similar, really. They mostly differ in the way that you set up the load parameters. But likewise, you specify collection of filtration to waste or collecting blank fractions. So it works quite similarly to the filter plate experiments in this context. And so let's take a closer look at how the load parameters work. So you had a brief glimpse earlier. It's really easy and really flexible because you can basically set up here what liquid you want to be loading, number of fractions, volume of fractions. And likewise, this is really a useful feature. You can specify the flow rate in either of these options, and the other two will automatically update. For the gradient chromatography, as I've alluded to, you simply need to specify which buffers you want to be using to generate the gradient and what is the number and size of the fractions for the stepwise gradient. And Synthace will take care of generating all the instructions to make these required intermediate mixtures. And so, again, going back to the example that has been percolating throughout this presentation, really, looking again at monoclonal antibody purifications, imagine you want to validate what you found in the filter plate experiment. So imagine you will want to maybe try loading different types of samples to see whether the findings from filter plates still hold true. So in this case, we were using 2 samples that had slightly different upstream process conditions. And we simply looked at the 2 different buffer types for the whole cell protein wash. And as I said, we fixed the elution pH to 3.6. And the goal, again, remains the same. We want to maximize yield and minimize whole cell protein impurities. And so once again, further slap in the face. Bam, what is this? It's, again, if you just take a breath and focus in, it's really simple because once again, it's very modular and it all flows quite nicely. And so you'll see here, you have define resins and RoboColumn plates. So these are the 2 elements I was mentioning earlier. This is simply you specifying, right, what am I dealing with? What's my RoboColumn plate looking like? And this is, again, simply a copy of that one element where you're specifying the different stages from all the way from equilibrations, load, wash, then the whole cell protein, wash, elute, strip, sanitize, recalibrate, and storage. Now you'll notice here I have some additional elements over here. And these are not specific to RoboColumns. They're just widely available in our platform. Here, I'm simply instructing Synthace to add a neutralization buffer onto the eluted fractions to protect the eluted protein from precipitation. And finally, we are reading all the plates automatically on the plate reader. What this means is, in practice, Synthace will automatically generate instructions to move the plates to the plate reader. So you really just set this up and leave. So you can imagine even like a 6-hour purification run, this can be quite handy. And so I'll just give you a brief preview of how it looks like, again, in the simulation preview. And similar to filter plates, you see exactly what will happen. And you can course correct very easily before you even go in the lab. And I hope you can appreciate this is much harder to do when you're scripting on the native software of these devices. But the point I really wanted to highlight, and that's really, to me, the biggest gift, is the data analysis app for this application. So imagine you've done your run. All you need to do is click on chromatography results. And in the first tab, you already have raw chromatograms available. So this is just the raw data plotted against the loaded volumes. But alternatively, you can choose also the fraction number, and you can select and deselect whichever RoboColumns you want, whichever wavelengths you want. But where the fun really happens is in the Analyze tab, as here you select the wavelengths you want Synthace to use to calculate bath length, what wavelength to use for background subtraction, and for protein absorbance. You can also optionally do blank correction, but you also then have the option to set up the K factors for the different buffers for your samples, and set up a molar extinction and molecular weight for calculating mass and molar concentrations. And so here, this is my favorite view. It's the plate view where you already have normalized data. So you see it's showing A280 at 1 centimeter. So this has been normalized to 1 centimeter path length. And again, it's the same pseudo-chromatogram graph, which you might be familiar with from ACTA purifications. And again, it's quite flexible. You can plot it against fraction number. And again, here's the same data, but in a plate view. And you can hover over the different points to see what the data is in the individual wells. Over here where we have our cherry pick app, but I will touch upon that later. And here is a sample data table, which captures the data that was just visualized up there, but in a table. So you have the raw data alongside the calculated data. And finally, in the cumulative data table, it aims to sum up the different rows to give you a better understanding of the overall result of the process. Obviously, all these data tables are fully exportable, so you can take them offline and work with them elsewhere if you so desire. And now for the cherry pick, here you can see I've selected the eluted fractions, and they became highlighted where they physically are on the plate. And I have simply decided, okay, I want to cherry pick and pull these fractions. So all you have to do is click Render Workflows. Our destination plate type here is 96-well plate. And so what this will do is generate a new Synthace workflow where all we're going to do is simply transfer the fractions from the original collection plate. Here we're defining what plate we want to go into. And then we hit Simulate. And once again, once the simulation is done, you get a preview of what the robot will execute. So here, it's simply taking the fractions and pulling them into a single well from each robot. So I hope that was a quite nice overview of how powerful Synthace platform can be when it comes to running either filter plate or RoboColumn experiments. But I think you could appreciate that RoboColumn experiment was quite simple. We weren't really looking at many factors. It was, quite narrow range. But to imagine you want to continue exploring that experimental space. And so you want to apply DOE. So let's quickly touch upon what is actually DOE and why you should be considering using this type of methodology for designing your RoboColumn experiments. So in this little diagram over here, what I'm showing is two different research groups. So research group A and research group B. They have both decided that they want to optimize a particular process. So let's say increase the yield of a particular protein. And for this, they have decided to vary factor 1. You can see that both of them decided to increase factor 1, but somehow found opposite results, where team A found that increasing factor A decreases the yield. Team B found that increasing factor 1 actually increases the yield. And so you might wonder, how is this possible? How can they have found such opposing results? Did one of the teams make a mistake? And the truth is, no, they did not. The simple reason here is that team A and team B decided to fix a second factor at a set level. So whereas factor A decided to set factor 2 at high level, team B has decided to set it at a low. And so then what has happened is that they have missed an interaction between factor 1 and factor 2, where actually there's two optima for the yield, but that is determined at what set point factor 2 is. This relies on varying one factor at a time. That's what OFAT stands for. And so I hope you can appreciate that with OFAT, sure, both teams have eventually gotten to the right optimal condition, but they really didn't understand much about their process. They didn't understand that factor 1 and factor 2 interact together. And so in a real life situation, they could easily get a bit caught out. But in contrast with DOE, what you do is you vary both factor 1 and factor 2 at the same time. And so the end result is you are screening less conditions at the same time. So you can see here by the number of points, we have 5 here, 5 here, so 10 in total. Here you've screened 5 as well, but you understand so much more about your system. And so how does this actually affect your process development? In other words, why should you care? And the simple reason is that you have to run many more experiments to understand your system. And at the end of the day, you still don't actually understand it. Because as you can see here, you go through the process of varying one factor at a time, where you end up fixing the factor at the optima that you found, which in this case, it's here. For this factor, for second factor, it's here. For third factor, it's here. But if you run a DOE, you can get to that place much faster and in a smaller number of experiments. And so going back to our monoclonal antibody purification examples, so imagine we wanted to be a bit more thorough and we want to explore more factors. So imagine a scenario where, okay, we've screened in our filter plate, we've used a particular type of resin, but in a column format, maybe things will be different. Let's try another type of resin. Likewise, the protein concentration that your sample is at can also influence maybe the amount of impurities you will see coming out at the other end. So here we'll probably try exploring 3 different protein concentrations. For the whole cell protein wash, again, we're still sticking with the 2 different buffer types. And we've narrowed down the range of the salt concentration that we want to try. But likewise, we think it's also important how much we end up washing the column. And finally, the elution might be influenced by how fast the liquid goes through the column. And of course, the goal still remains the same. And so to do this, you might end up using a DOE design software such as Jump or Design Expert to actually generate a DOE design that can satisfy learning about this kind of experimental space. As you can see here, this is the design that we came up with, where we are looking at these different factors. And now you're thinking, oh, how am I going to set this up in Synthace? Do I need to do it one by one? The answer is no. So to start with, what you need to do is design what we call a pilot workflow. So what you do here is you simply set the different parameters you will be exploring, and you're trying to set them to high and low for every single parameter. And this is to basically ensure feasibility. Because if your parameters are, your level that is high somehow cannot be satisfied based on the Synthace calculations, then obviously the whole design is not feasible. And so going straight to DOE design doesn't make much sense. So here, all we're doing is checking these factor ranges are feasible. And so we simulate. And so you could see, hopefully very quickly, that simulated correctly. But next, what we actually have to do is import the design that I've shown you in the Excel file into Synthace. And so to do this, we have this toggle switches over here where we simply parameterize which parts of the process we are looking at DOEing, if that is a word. And so you see here, we're setting up the protein concentration and the levels for it that we're going to be exploring. So we want to have 1 mg per mil. This is what the design has dictated to us, 4.5 and 8 milligrams per ml. This part looks a bit scary, but it's simply we're letting Synthace prepare all the protein that will be loaded. So we're letting it dilute it. And also for the wash buffers, we're letting it prepare the wash buffers with the added salt so that we don't have to worry about calculating all this stuff manually. And so again, here we're parameterizing the wash buffers that we're going to be using. And again, here in the whole cell protein wash, we're going to be changing the load volume per fraction. So this will vary between 0.5 column volumes up to 1 and 1.5. And so I hope you get the idea of the stages that we go through. So you simply need to tell Synthace, these are the factors I want to be looking at. These are the ranges. Let me jump forward. And then we get taken to an experiment design interface. And so here you get a summary of the factors you will be exploring. Here we're adding one additional factor, which is the random block. This is just part of the design that we've created, where we decided to split the design into 2 separate blocks. The idea being it's 32 runs, so it's 32 columns. We just want to speed things up and maybe run them on separate days. And so you see here, we're referencing this file, which we need to upload into Synthace so that Synthace knows what should be the set points, what should be the combinations of the factors for each specific RoboColumns. And so all we're doing here is we're matching what we set up in Synthace with what is in the Excel design file. So this is so that Synthace knows how to interpret the file that we're importing in. And once we're happy with that, we simply need to tell Synthace that we are interested in splitting up the resulting workflow. And so here we simply need to click render, and that's it. That's our workflows ready to go. And so here I'll quickly showcase you how this looks like. So again, we get taken to the builder and you see the workflow looks exactly the same, but now you can see that all the parameters are automatically filled in. So you didn't have to do this manually. Synthace imported this from the design file. And that there, it's basically columns 1 all the way through to 16. And we're highlighting here the different stages that we decided to DOE. And this is the second workflow. So as you remember, we split the design into 2. And again, here we're showing the remaining portion of the RoboColumns. And again, all you need to do is simulate, and that should be ready to go. And then once again, you can go into preview and check everything that has to happen. Once again, you get given exactly what buffers you will be needing to run this, and so that you can plan ahead of time. And with this, I'll hand over to Victoria.
Thank you, Lucy. So hopefully today we have been able to show you how it really is possible to explore your downstream processing design space very effectively with different high-throughput process development tools, and empowered by Synthace, you can make those experiments more insightful, since we're helping you with preparation, visualization of data for analysis automatically so that you can access to those insights faster. Hopefully also enable you to build more powerful experiments, so build and simulate experiments with our embedded design of experiments tools that help you to design, iterate over your workflow troubleshoot before even setting foot in the lab, and then we take care of the execution as well. And finally, make your experiments more reproducible. So we generate those interoperable device instructions as you go, as you are designing, that can then be run on the device so that you can focus on the science, not the feasibility, and that you can easily transfer these methods, these scripts with your other colleagues, different teams, different devices. And here are just some numbers that hopefully speak for themselves in terms of the impact that we've had so far when purification teams have used Synthace. So this is with miniaturized purification with RoboColumns. But as we've shown you today, we now also enable miniaturized purification with filter plates, which we're very excited about. The type of effect that we see is really a big increase in the number of scientists that can now use liquid handlers and actual device utilization time, as well as time savings for the average experiment in terms of how long it takes people to design and particularly to wrangle data, as around 80% is what we've seen in terms of increases of data processing compared to manual data processing. With that, we'll conclude today's webinar on downstream process optimization. And if you're at all interested in learning more about downstream process optimization or other applications with Synthace or what it would look like in your lab, do let us know. All right. Thanks, everyone, and have a great rest of your day or afternoon, depending on where you're tuning in from.
Thanks. Bye.